That's probably good. Probably good. In this day of violence in the classroom. I'm sorry?
We did it, and then you were like, that's bad.
Well, you guys don't do everything I tell you to do. So, you try to be good. Oh. They're hissing me. Okay, so. All right. No, we won't do that. But I'll give you a hard time, okay? It is kind of cute, though, isn't it? I mean, it's like... Okay. All right, enough. All right. So, it is Friday. We are all going to be supportive of each other. We are all going to join hands and sing Kumbaya, right? No. Jeremiah was a bullfrog. You know, I don't want to get into starting this any more than you guys do. We should just stand around and just talk all day. It'd be kind of fun, wouldn't it? There's a vote for that? I hear a vote for that.
How are you feeling today?
Well, thank you for asking. I'm feeling like I'd be happy to do this, but you guys wouldn't like the fact that I'd have to go twice as fast at the next lecture to make up for everything.
How does that make you feel?
How does that make me feel? I can talk fast. Okay. How was the run? I didn't run this morning. I ran yesterday morning. Had a great run yesterday morning. And I'm running a 10K tomorrow. Anybody wants to run with me? Come running with me. I'll be out there running. We meet at my house and take off about 9 o'clock. So come by. What's that? I am going to do the half marathon in April. Anybody want to run with me? You're doing it? Okay. How many are doing the half marathon in April? 1, 2. There's A's right there. Okay. The half marathon is 13.1 miles. It is the first Corvallis half marathon, and it is running through all over Corvallis. And I myself have never done a half marathon in my life either, so it'll be. Maybe you guys good to get to know me while you can. I may not be here. I may not survive that, you know. So maybe it's bad to get to know me now. I don't know. But the half marathon will be fun. So, all right. Should we get started? Now. Happy Friday. We're back where we started. All right. So we have an exam in here on Wednesday. I haven't decided where to cut things off. I will announce that later. We're obviously not cutting it off today. So it'll be some point at the material on Monday. Oh, I know. Yeah. I did upload to the website a practice exam. Everybody should look at that practice exam and read the instructions. Okay? It's a different format than last term. The instructions will be exactly the same. The format will be exactly the same as this term, although the number of questions in each section may change a little bit. But the question will be the same. The first section, people frequently confuse it for a multiple choice. It is not. So read the instructions, because I always hear from 1 or 2 students after they've taken the exam, I thought it was a multiple choice. It's not. So make sure you read through that first section and the instructions in there, okay? All right. Last time I introduced you to respiratory control, and I hope that you found that topic as interesting and as intriguing as I do. I think that respiratory control explains so many things in our bodies with respect to energy, with respect to metabolism, with respect to what we eat, with respect to how we get fat, and all of those sorts of things. And so the explanations with respect to exercise and energy and obesity and so forth make a lot of sense. So I will just very briefly remind you of a couple of things with respect to that. One is that ADP is a limit if we're not exercising. ADP is necessary for complex 5. If complex 5 does not run, and I hope, again, when you think of respiratory control, you start thinking of all these connections that I'm giving you here. You should be able to do these in your head like I'm giving them to you. All right? What if I said to you, you have no ADP? I'd like you to think about all the possible things that would mean. No ADP means complex 5 is not turning. Complex 5 is not turning means gradient is not going down. It means that the complex is trying to pump protons will have to stop because the gradient is too large. When the complexes stop pumping protons, they stop flowing electrons. No electron transport, NADH levels go high, FADH2 levels go high, citric acid cycle stops. Okay? Citric acid cycle stops, acetyl-CoA concentrations go up. Acetyl concentrations go up, we start making fatty acids. We'll be talking about that on Monday. Acetyl-CoA is necessary to make fatty acids. Somebody asked me at the end of the last period, well, what happens when I run out of oxygen? Okay, good question. Let's think about what happens when I run out of oxygen. I put a bag over my head and tie it, and I don't have any oxygen. It will kill me. Why does it kill me? It kills me because of the following. Oxygen is the terminal electron acceptor. Right? No terminal electron acceptor, no electron transport. No electron transport, what happens to NADH concentrations and FADH2 concentrations? Up, right? Now, I hold that bag on my head long enough, I'm going to die because I have no energy. Okay? Now that's different than the situation we had when we talked about your muscle cells running out of oxygen, because your muscle cells had something that saved them. What was it? The Cori cycle saved them, right? Because the liver was able to deliver glucose to them to keep them going so they could do fermentation. Okay? You can't stay alive with fermentation keeping your liver alive. Your liver needs to be able to do all these things that the liver is so good at doing. If it doesn't, you're going to die. And that's what happens if you put a bag over your head and suffocate. Okay? So, all these things are interconnected. We're going to see even more interconnections when we get to fatty acid metabolism, so I'll save those for you. But I want you to understand the basic principles that I have given you about these different processes here. Okay? Are there questions about these? A scenario you'd like to think of, maybe? Okay? So think about what happens when we have coupled versus uncoupled. Coupled involves intact mitochondrial membranes. When things are coupled, electron transport and oxidative phosphorylation are interdependent. If one stops, the other one will stop if we have coupled mitochondria. If we have uncoupled mitochondria, they are no longer interconnected. We uncouple mitochondria, we stop making ATP. Because what we do is we poke a hole in the membrane, and protons no longer move through complex 5. Protons come in through the hole. In that case, what happens is we stop oxidative phosphorylation, but electron transport goes like crazy. That was the diet drug idea, right? Electron transport's going like crazy. Oxygen consumption goes like crazy because electron transport's going like crazy. Nothing to stop it. It keeps going, and eventually you burn things out. One of the things I mentioned briefly when I talked about 2,4-dinitrophenol, which is this compound that uncouples mitochondria by poking a hole in it, is that the person who took this would probably breathe pretty heavily in their sleep before they died. They would probably be hot, okay, because they're going to be generating a lot of heat. And it turns out, surprisingly, that there are biological processes that are involved in uncoupling as well. Okay? Now, you might think uncoupling, whoa, bad, okay? But in fact, uncoupling turns out to be very beneficial in some cases. And I'm going to give you one case where uncoupling is very beneficial. Okay? It actually relates to this thing at the bottom called uncoupling protein. Okay? We have in cells of our body called brown fat, we have a situation that looks like this. Uncoupling protein is a protein in the membrane of the cells of brown fat. Brown fat, we don't have a lot of it, but where we have it in our bodies is located around our spinal cord. Okay? Now, what happens is uncoupling protein, when it opens up, it will allow protons to move through it. It's not open all the time. It's only open part of the time. And that's the key to its function. Uncoupling protein opens up and lets protons move through it when the cells get cold. When the cells get cold, protons move through it. We do not make ATP, but we generate heat. This uncoupling protein is generating heat in the area near our spinal cord because transmission of neural information is, in fact, dependent upon temperature. Because why? It's dependent upon diffusion. We would like to be able to have our nerves and our responses working well even if we are very, very cold. Uncoupling protein allows those nerve cells to get a little bit of heat because of the movement of protons and the fact that we're speeding everything up. You had a question?
Yeah, this is in the mitochondria of brown fat.
Mitochondria of brown fat cells. I'm sorry, yes, right.
How cold would you have to be for that to happen?
How cold would you have to be for that to happen? I don't have a temperature. I can't tell you. But suffice it to say you would have to be fairly cold. Yeah. Infants have more brown fat cells than adults do. And different organisms have more brown fat cells than humans do, not surprisingly. Not everybody lives in an air-conditioned, 70-degree constant temperature environment like many of us do. Okay, makes sense? So, do you see why uncoupling protein generates that heat? It does because it's uncoupled, so everything starts running. Electron transport runs like crazy. Citric acid cycle runs like crazy, and all that metabolism, the byproduct of it is heat. Okay, makes sense? Yes, question back to that. You know, I honestly don't know if brown fat is called that because it's physically brown or not. I think so, but I don't know that. Okay. Well, the last thing I want to say about oxidative phosphorylation is a set of considerations. And that was up here with respect to the mitochondrial membrane shuttles. So I want to say a word about that. I haven't told you something. The something I haven't told you is that NADH and NAD do not cross the mitochondrial inner membrane. They do not cross the mitochondrial inner membrane. The NAD and NADH that are in the matrix are there because they've been made there by the mitochondria. They didn't get there by movement across the membrane because there's nothing that will move them directly across the membrane. Okay? Well, that poses a little bit of a problem because if you recall, out in the cytoplasm we have glycolysis going on, and glycolysis in the cytoplasm is generating NADH. So we sure would like to be able to use the electrons from NADH in the electron transport system, but to do that, we've got to get those electrons in. Well, if we can't move NADH in, we have to figure out a way to move the electrons in, and that's what I'm getting ready to tell you about. The electrons come in by means of what are called shuttles. Okay? The first one I will show you is very simple. It occurs in insect muscle. Where did I mention insects recently? Rotenone, right? What was rotenone doing? It was an insecticide. And I said insects were more susceptible to it. I hope after I finish this you'll see a little bit or have some idea about why they might be more susceptible to it. Okay? Insects use this system that you see on the screen. They produce NADH out in the cytoplasm. The NADH transfers its electrons to dihydroxyacetone phosphate and make glycerol 3-phosphate. Notice that's glycerol 3-phosphate. Glycerol 3-phosphate interacts with a protein in the mitochondrial inner membrane. That's the FADH2 generating protein. Okay? This was succinate dehydrogenase. It's a very flexible enzyme. This guy drops off its electrons with this protein and makes FADH2. FADH2 enters the electron transport cycle and we go on, right? The byproduct of dumping the electrons off is dihydroxyacetone phosphate, and so we've done a complete cycle. The problem is we started with NADH, and down here we have FADH2. What was the difference between those two compounds in the electron transport system? Okay, so complex 2 handles electrons from FADH2, right? And complex 1 handled electrons from NADH. Which one generated more ATPs? Which one pumped more protons first? Okay, so if you go through complex 1, you pump more protons. If you come through complex 2, you don't pump as many protons, you don't make as many ATPs ultimately, right? You don't charge the battery as much. right? Now, you've just lost some energy. The advantage of this system is it's very, very fast. Insects need to do things fast. They need energy fast. They trade off speed for efficiency. This is not the most efficient, but it's very, very fast. Energy is at a premium for insects because of this. And now you start to see why rotenone makes them much more susceptible, because rotenone is blocking electron transport and they're needing that energy as much as they possibly can. Okay? Everybody got that? So this is a shuttle system. Yes, Shannon.
Rotenone blocks complex 1, right?
Rotenone blocks complex 1. That's correct.
So they have more FADH too, so wouldn't it be more effective with block complex 2?
Well, this is only true for things out in the cytoplasm. Remember, they make a lot of NADH in the matrix. And if you block complex 1, they're not getting any of those NADHs from the matrix either. So they're much more, it's a good question, but they're much more susceptible to energy loss as a result of that. So by blocking complex 1, they stop the NADH from the matrix from working as well. Okay. Now, as is always the case in biochemistry, there are simple systems and there are more complicated systems. Human beings, of course, are much more complicated things than insects are, and so we get to have a fancier system. Yay us, we're number 1, right? Okay, we are number 1. That's right. Now, we use something called the malate aspartate shuttle. All right? And this one looks a lot hairier than it is, and it's actually very straightforward, especially the way we're going to look at it. All right? The malate aspartate shuttle involves, as its name suggests, malate and aspartate. This figure is rather stupid in several respects, but one respect in which it's stupid is it looks like things are going this way and they're going this way, and in fact, that's exactly right. It's kind of dumb to do them both the same way. Why didn't they put NADH up here and then NAD over here so they both go the same direction? I don't know. The only important thing here is that the big reaction with respect to electrons that's occurring is occurring right here. Okay? It's occurring right here. We have oxaloacetate is gaining electrons and becoming malate. That's the reversal of the reaction we saw in the citric acid cycle. NADH is dropping electrons off into oxaloacetate to make malate. Now, there is a shuttle that will move malate across the mitochondrial inner membrane, and so it moves malate in. When malate gets into the matrix, the reversal of that previous reaction occurs. Malate gets converted back to oxaloacetate and makes NADH. So, this is the entire process in essence. All right? Oxaloacetate is going to malate, malate is going to oxaloacetate. NADH going to NAD, NAD going to NADH. We've just moved electrons into the mitochondrial matrix, and we haven't lost anything. We haven't given up any energy in that process. This is much more efficient, but it's slower. Okay? Well, what's all this other stuff on here? Well, all this other stuff is just to make the process a cycle. Okay? All that really matters here is that we've gone back. We've now got oxaloacetate. Oxaloacetate can go to aspartate. Aspartate can go out. To get oxaloacetate to aspartate, glutamate has to go to alpha-ketoglutarate, and it goes back out. Okay? The important thing here isn't all the details of who goes what where, but what's being used. That's what matters. Malate and aspartate. Malate and aspartate are used to shuttle electrons in. Malate is the carrier of the electrons across the membrane. That's the bottom line. Okay, questions on that? You guys are a quiet group today. We've started off on the right foot, I guess, maybe. Or the wrong foot, I don't know. Yes, question?
Is it okay to draw it the other way on the top, have the arrow going the other direction?
As long as you're showing what's happening. Yeah, it's just a dumb, why they do it one way here and then a different way down here. It'd be nice if they were consistent.
Okay.
Yeah. Yes?
This happens every time before electron transport, to get the NADH in.
Right. So this happens every time electrons need to come in from the cytoplasm. Remember, this is only in the cytoplasm to get these electrons in the cytoplasm. If NADH is already in the matrix, which it'll be for the citric acid cycle, and as we'll also see for fatty acid oxidation, if NADH is already there, this isn't even necessary. So this is necessary when we generate NADH in the cytoplasm, such as we do in glycolysis. Okay? Very important point. Shannon?
A little off topic, right? But NADH is fixed in the mitochondria. It's also fixed in the cytoplasm. The
Yes.
driver of NADH.
That's right.
So when a cell divides, does it make more NADH?
So the question is, you've got a fixed amount of NADH in the cytoplasm and a fixed amount of NADH, or NAD, either one, in the mitochondria. And when the cell divides, what happens? Obviously, the cell does, in fact, need to make more. But the cell will control how much total NAD that it will make. And that's true both in the mitochondria and in the cytoplasm. But yes, you're right. It has to make more when it divides. Okay. Let's turn our attention now then to, and I think we are basically done with all that. Bunch of transporters, blah, blah, blah. We don't need to worry about those. For those people who like counting things, your book counts 30 ATPs starting with glucose. I count 38, but again, that depends on how you count things. So we know we generate a lot more ATPs if we oxidize things with oxygen than if we don't. If we do fermentation, we run a lot shorter. Okay, let us turn our attention now to fatty acid oxidation. Okay, and I guess we're not doing fatty acid oxidation. What did I do here? No, I've got the right thing. That's how in my head I had myself doing fatty acid oxidation. I guess because I was thinking fats up here. All right. So we are oxidizing things that involve fatty acids, but we're not doing fatty acids themselves. That screws up the song I was going to do today. We'll have to save the song. All right. Then this actually saves us a bit, because this is where we will draw the line for the exam. When I finish this material, that will be the end of the material for the exam. Okay? That may take us into Monday. I'm not going to finish it today, probably. All right. Well, we've talked about the structure of glycerol phospholipids. We talked about the structure of sphingolipids. Now we're going to say a little bit about how these guys are made and how they're broken down. Okay? These are metabolic processes, and we need to understand a little bit about that. Okay? The synthesis of glycerol phospholipids obviously has to involve glycerol. And a starting point in that synthesis, this is one starting point. There's a couple of ways of starting, and we're not going to go through all the different ways. We're going to start, in this case, with glycerol 3-phosphate. Glycerol 3-phosphate you saw out in that shuttle process, so there's a way of making glycerol phosphate if we need to make it for some reason. Glycerol 3-phosphate can become esterified to two fatty acids. Remember, this is 1, 2, 3, where 3 is the phosphate. 1, 2, 3, where 3 is the phosphate. The first fatty acid to get esterified is on carbon number 1. Becomes linked, as you see here. It creates something called lysophosphatidate, which we won't care about the name. And then a second fatty acid gets attached at position number 2, and that creates a molecule called phosphatidate, or as we more commonly call it, phosphatidic acid. Phosphatidic acid is a glycerol that has 2 fatty acids and a phosphate attached to it. You'll notice that fatty acid number 1 is usually a saturated fatty acid. Fatty acid number 2 is usually an unsaturated fatty acid. You start to see why unsaturated fatty acids are essential fatty acids. We have to have them to make glycerol phospholipids for our membranes. Okay? All right. Phosphatidic acid turns out to be a branch point for the 2 different pathways. One pathway is making the phosphatidyl compounds, phosphatidylethanolamine, phosphatidylserine, phosphatidylcholine. These compounds are made from phosphatidic acid. Phosphatidic acid can also be used to make fat. So it's a branch point between the synthesis of the phosphatidyl compounds and the synthesis of fat. Both of them have phosphatidic acid as their intermediate. Okay? If I want to go to fat, I have to take phosphatidic acid and pull the phosphate off of it. Then I make this interesting compound called diacylglycerol. And where did we hear about that before? In the signaling process, right? IP3 signaling, exactly. And that's what we see here. Here's diacylglycerol. This could be a signaling molecule. It could also be used to make some fat. And we do that by taking the third carbon, attaching a fatty acid to it, and bang, we've got a triisoglycerol. Now, fat is not stored by all cells. Fat is stored by specialized cells of the body called adipocytes. And these can get really full of fat. The reason being, fat is not water soluble. Glycerol phospholipids can exist in the membrane because they can associate with water and associate with each other, et cetera, et cetera. Fat can only be nonpolar. So it's not surprising that we keep fat stored in specialized cells that are capable of handling this very nonpolar compound. Okay. Now, how do we make the phosphatidyl compounds? Again, there's more than one way to do it, more than one way to skin a cat. We're not going to go through all of them. But one we will go through is right here. Okay, here's phosphatidic acid. Phosphatidic acid, as you will see, is becoming linked to cytosine diphosphate. Okay? Cytosine diphosphate. That's CDP. That is a nucleotide. And the last time we linked something to a nucleotide, we gave it a name. Not the name of the molecule, but we described it as an activated intermediate. Had a high energy bond. The last one we talked about was UDP glucose last term. An activated intermediate, you recall, has high energy and uses part of that energy to donate a part of itself to something else. And we'll see that happening in a minute. CDP diisoglycerol. All right? Now, what you've seen now is you've seen UDP involved in a high energy intermediate in glycogen metabolism. Now you see CDP involved in metabolism relating to glycerol phospholipids. Okay? We see ADP as a component of many things. We saw it in riboflavin. We saw a bunch of different things last term that ADP could be linked to. And GDP doesn't get always linked to something, although in some organisms it does. But GTP is involved in making protein. Now, I point this out to you because all four nucleotides play roles in metabolic pathways besides making RNA and DNA. They play roles in that. You need to have CDP to make this stuff. You need to have UDP to make glycogen. Okay? That means, therefore, that the levels of these nucleotides will be influenced by the processes that we see here. They'll be influenced by making of glycogen. They'll be influenced by making of glycerol phospholipids. They'll be influenced by the making of protein. Okay? It turns out that all these processes are pulling from that pool, and if they pull from that pool, they affect the levels of those things. When cells have to make decisions, do I divide or not divide, they have to know, do I have enough things to make not only my RNA, my DNA, but also these things here. The integration of these pathways gives cells immediate answers about what do I need to do. Do I have the energy to do it? Do I have the resources to go on and do my thing? Because all four are involved in other metabolic pathways. Okay, well, I'm getting off subject. CDP diisoglycerol, as you can see here, is an intermediate, and we take CDP diisoglycerol, and we can make a variety of other compounds. Basically, in this case, if we take the CDP diisoglycerol and we combine it with inositol, we can make phosphatidylinositol. This guy, this portion leaves, the portion in blue, one of the phosphates stays behind, and we've got this compound. CMP, as a result, comes off. I could do the same thing with serine. I could do the same thing with choline. I could do the same thing with ethanolamine, and I could make phosphatidylethanolamin. All I have to do is substitute serine, choline, ethanolamine right there, and bang, I've made the equivalent phosphatidyl compound. So the synthesis of phosphatidyl compounds metabolically is not overly complicated. No, we're not going to draw all those structures. Here's ethanolamine. And there's other ways of doing it. You see some of these as are shown here. In this case, they take the ethanolamine and link it to CDP. The end result is the same. You make phosphatidylethanolamin, so you can mix and match these to a large extent. Okay. Phosphatidylcholine is an interesting compound, and I bring it up here because we'll talk about this a little bit later. Phosphatidyl ethanolamine is a precursor of phosphatidylcholine. So here's the phosphatidyl part way out here. Here's the ethanolamine part out here. All right? When we look at this, we see that there's a difference between the phosphatidylethanolamin and the phosphatidylcholine of 3 methyl groups. Those 3 methyl groups get there by action of a methyl donor. First time we talked about a methyl donor. A methyl donor is a molecule, not surprisingly, that donates methyl groups. It donates 1 carbon. It's part of what's called 1-carbon chemistry. It donates 1 carbon at a time in the form of a methyl group. 3 of these go to make phosphatidylcholine. Now, it turns out 1-carbon chemistry is interesting and important. One of the intermediates involved in that is the one you see on the screen. It's called S-adenosylmethionine, or SAM. You can call it SAM, S-A-M. After it's donated as a methyl group, it's converted into S-adenosylhomocysteine. And this we'll say more about later. I think I'll save that for until later to talk about. Okay? But there are 2 big systems our body has for donating single carbon groups. One is the S-adenosylmethionine system, which you see here. The other system is called the folate system. The folate system turns out to be very important for nucleotide metabolism. Okay, so we just see a little bit of chemistry going on here. Sphingolipids. Sphingolipids turn out to be synthesized fairly straightforwardly also. Sphingolipids look like they come from sphingosine, but technically they don't. We sort of cheat on this. We say, oh, the sphingolipids are all related to sphingosine, as if they come from it, but they really don't. Sphingosine looks like the sphingolipids. But in fact, the sphingolipids come from a fatty acid and an amino acid. Okay? A fatty acid and an amino acid. The fatty acid is palmitoyl-CoA, palmitic acid, basically, plus serine, when you link them together, create a sphingosine-like molecule. Okay? We got these big names. We're not going to worry about the names. All right? There's several rearrangements that happen, and we eventually get something called a ceramide. All right? So there's a term we haven't heard, a ceramide. A ceramide is a sphingosine-like molecule. If we leave it with that name, that means we won't have to worry about all these other things. A sphingosine-like molecule to which we've attached a fatty acid. Okay? The fatty acid was actually attached right here, but at this point, you see this is a dihydroceramide. All right? So a ceramide is a sphingosine molecule that has a fatty acid that's been attached to it. So we've gone from a sphingosine-like molecule to a ceramide by adding a fatty acid. We made the sphingosine-like molecule by starting with a fatty acid and serine. All right? That gave us this. We added another fatty acid over here, and we've got a ceramide. If we want, so there are in total 2 fatty acids. One that was involved in making the sphingosine-like molecule, and one that is used in making the ceramide. If we take that ceramide and we now attach out here something else, we create something we think of as a sphingolipid. This something else could be phosphatidylcholine, in which case we made sphingomyelin. This could be a simple sugar, in which case we've just made a cerebroside. This could be a complicated set of sugars, in which case we call it a ganglioside. You've seen these structures before. Now I'm just briefly telling you how we make them. You guys look very glum today. After that really early banter, we got off on a good foot, and now everybody's just worn out. Their smile muscles have all worn out, and they're just looking very glum. What's that? You'll smile? Okay. I have a dumb joke if you want a dumb joke. Actually, I have an even better joke. I've never told this joke before. I just read it yesterday. Okay? So let's see if I get it right. Okay? So this guy is standing in line at the grocery store. Okay? And the guy in front of him has a bag, a 50-pound bag of dog food. Okay? And there's this lady behind both guys, and of course she's one of these chatterboxes, right? And she walks up and she points at the bag of dog food to the guy, and she says, "Do you have a dog?" So the guy in the front says, "Well, ma'am, as a matter of fact, no, I don't. I buy this dog food because I've been thinking about going back on this diet that I discovered, the dog food diet." Okay? And the dog food diet, he says, before, when I went on the diet, I thought it was really great. After I was on it for a while, next thing I knew, I woke up and I had these tubes coming out of me, and I was in the hospital. But it's really a great diet. She looks at him and she says, "What do you mean?" He says, "Well, here's how it works. You got this dog food, and I fill my pockets with the dog food. And every time that I get hungry, I just reach in, I grab one of these pellets of dog food, and I eat it." He says, "Nutritionally, it's okay." He says, "And it doesn't taste bad, and it keeps me from eating a bunch of other stuff." He said, "I lost 50 pounds doing this thing." The lady says, "That's really great, but is that why you ended up in the hospital, with all the tubes and everything coming out of you? Was it the diet?" He said, "No." He says, I was walking along, I went to smell the butt of a poodle, and a car hit me." Okay. I thought it was better than that. You see, it was eating dog food. It kind of snuck up on you, didn't it? I didn't say that one right. I should have been more careful. No, the other one I'll save because you're clearly not in the mood for a good joke today. All right. This shows you on the screen what I told you in words a minute ago, before the poodle joke. And that was, poodles are supposed to be very smart dogs, too. Did you know that? Anybody have a poodle? Are they smart? The what?
Not the red kind.
Not the red kind. Do you dye it or anything?
No.
No.
It's just a special brand, and they're supposed to be like the ADD poodles.
Oh, really? So stay away from the red poodles.
Yes.
Anybody else have a poodle? Are they smart?
Extremely stupid.
I've always heard they were smart.
They are smart. They're just clever. They're
Smart,
smarter
but
than you.
well, that wouldn't take much, but me, that is. Okay. Here's the ceramide. Okay. Structure, blah blah. We attach a phosphatidylcholine to it. We get sphingomyelin. We attach a simple sugar to it. We get a cerebroside. We attach complicated sugars to it. We get gangliosides. Okay. Let's see. Gangliosides, we can schematically see, are rather complicated. A lot of different things that can be out here. It's not one sugar. If it's one sugar, we've got a cerebroside. If it's many, we've got that. Okay. Now, there is an enzyme called Tsachs Disease that is a very debilitating neurological disease. And it arises from the inability to break down gangliosides. Okay? A specific ganglioside. In fact, what looks like a fairly minor ganglioside is at a fault here. Okay? Tay-Sachs disease is a very debilitating neurological disease, and it has an enzyme, a defective enzyme at its root. Okay? Okay. I won't say much more about that. There are quite a few diseases, genetic diseases, that are linked to defects in the ability to break down and recycle gangliosides. There are many of these. Because as you can imagine, there's a lot of enzymes involved in putting each of these individual things on. And there is a recycling process where they have to also be taken off if we want to make something else. Defects in those processes are not uncommon, and they do have some very, very severe effects. Okay.
Kevin?
Yes, ma'am.
Because peptides and gangliosides are most prominent in nerve cells,
Yes.
those side effects tend to have neurological.
That's exactly why they're neurological in their root, is because they are occurring in things mostly in the brain or in nerve cells. Yep. Now, Tay-Sachs is an odd one. Tay-Sachs, have I described this before? No. Tay-Sachs is a disease that if you have it, people who have it chew their lips off. They do. They will chew the ends of their fingers off. They will chew, chew, chew. And if they're not restrained, they will actually chew a hole in their mouth like that. Yeah. Is that right? Yeah, well, that's when it's discovered in babies, obviously. Yeah, they don't live very long. But they have to be restrained because they will put them and just eat the whole thing. Yeah. Weird. Okay. We've got about 8 minutes. Let's turn to our friend cholesterol. Now, we're going to focus for the moment on how cholesterol is made. The biggest considerations in cholesterol aren't how it's made, but how it's moved in the body. And that's one of the things I will talk about later. And that's also a lecture that I get a lot of questions on. So I won't talk about that today. I will get to that later. All right? Today I want to talk about how cholesterol is made. And cholesterol is made in a very interesting way. It starts with very, very simple compounds. In fact, we can make all of the carbons in cholesterol. We can put all the carbons in there using only acetyl-CoAs. Every carbon in cholesterol gets there from acetyl-CoA. Obviously, it takes many acetyl-CoAs. You can see the red carbonyl carbon. You can see the blue methyl carbon. And now you can see the location of every one of these in this cholesterol molecule. Okay? Oh my God, this is going to really be tough. Well, there are actually some very nice ways of simple fates of this. Okay? Here is acetyl-CoA. Acetyl-CoA we made by breaking down pyruvate. We'll talk later about how two acetyl-CoAs can be put together during fatty acid synthesis. They can make something called acetoacetyl-CoA. Okay? If we take an acetoacetyl-CoA and an acetyl-CoA and we join them together, we make something called HMG-CoA. Okay? HMG-CoA, this molecule right here, is known as HMG. I'm sorry, I back up. This guy right here is a 6-carbon molecule. Okay? And this 6-carbon molecule has two fates. One fate is that we can use it to make something called ketone bodies. We'll talk about that later in the term, and I'll remind you of this figure when I do that. But for our purposes today, HMG-CoA goes to a compound called mevalonate. So two different enzymes, two different directions for the same molecule. Again, this is a branch molecule. It goes to two different things. If we go in the upwards pathway, we go in the direction of making cholesterol. So if we want to make cholesterol, we go up. And the enzyme that's involved that catalyzes that conversion of HMG-CoA into mevalonate is known as HMG-CoA reductase. HMG-CoA reductase. You notice there's some NADPHs involved in this process. HMG-CoA reductase turns out to be a very important enzyme. HMG-CoA reductase is the regulated step in the synthesis of cholesterol. It is feedback inhibited by the end product of the pathway, which is cholesterol. Cholesterol feedback inhibits HMG-CoA reductase. When cholesterol starts to build up, the enzyme becomes inhibited. Okay? Well, we know cholesterol is a health problem. Why don't we shut off HMG-CoA reductase? Okay? Well, cholesterol metabolism, as we'll talk about later, is much more complicated than simply the synthesis. Diet, storage, and synthesis. Three things to think about with cholesterol. It turns out the one we have some control over is synthesis, not only by feedback inhibition, but also by chemical means. How many people know anybody taking statins? Anybody ever heard of statins? Yeah, okay. So statins are drugs that are given to combat cholesterol. They're very, very effective, and they work by inhibiting HMG-CoA reductase.
Allosterically?
Allosterically, the answer is no. This is an artificial compound, okay? And this artificial compound looks like HMG-CoA. It looks like HMG-CoA. It is a competitive inhibitor of HMG-CoA reductase. It looks like it. Okay? Now, when you have people who have too much cholesterol, the first thing a doctor will try to get them to do is alter their diet so their cholesterol levels go down, their cholesterol consumption goes down, and hopefully their cholesterol synthesis goes down. But if they can't control it with diet, the second thing they will do is they will say, well, maybe we'll stop. I really don't like that, you know. Maybe we will stop synthesis. Okay? And when you stop synthesis, after you've tried the other things, it seems to work fairly well. Okay? All right. Now, I've got a couple minutes, and I'm going to start this, and I'll finish it next time. But I need to get from mevalonate to making cholesterol. Okay? Mevalonate has 6 carbons. One of those carbons gets pulled off, and we make a 5-carbon molecule. Okay? 5 carbon molecules are called isoprenes. Okay? Isoprene. See 2 double bonds there. And isoprene is not the name of one molecule, it's the name of a class of molecules. We'll see there are 2 isoprenes involved in synthesizing cholesterol. Okay? One of these is known as isopentenyl pyrophosphate. Okay? Isopentenyl pyrophosphate. If you want to call it IPP, I don't mind. I'll count it wrong in the exam, but I don't mind. Okay, all right, so mevalonate gets converted to isopentenyl pyrophosphate. This is an isoprene. Okay? Several steps. We don't really care that much about the steps. The most important thing is we've gone from 6 carbons over here to 5 carbons over here. We've decarboxylated it. Isopentenyl pyrophosphate is readily converted into another isoprene called dimethylallyl pyrophosphate. And I don't have a name for that one. Was it Virginia? Was that what we decided?
Yes, I remember Virginia. I had bibs.
I like Chelsea. Okay, well, we'll call this Chelsea. Okay, so we can call this either dimethylallyl pyrophosphate or Chelsea. Okay, I'm sorry, but I'm the professor and I can call it anything I want to. All right, no. All right. Now, so we have 2 isoprenes. These 2 isoprenes are the building blocks, or the 5-carbon building blocks of cholesterol. Okay? Acetyl-CoA was the ultimate building block. They were used to make these. These guys are now used to join to make cholesterol. I've got 1 minute, and I'm going to show you how to make cholesterol in 1 minute. How do we do it? It's actually pretty straightforward. Not using that one, it's not. Okay? It's a dumb figure. Basically, we take one Chelsea, we take 1 IPP, and we make a 10-carbon molecule. 5 plus 5 is 10. Geranyl pyrophosphate. If we add, that's why this is kind of dumb here, if we add a 3rd isoprene to it, we get a 15-carbon molecule called farnesyl pyrophosphate. So we've taken either 2 5-carbon molecules to make geranyl pyrophosphate or 3 isoprenes to make farnesyl pyrophosphate. Okay, this figure, I pulled the wrong one up, is not the one I wanted. This is what I wanted. Okay, dimethylpyrophosphate, which is Chelsea plus IPP, makes geranyl. Geranyl plus IPP makes farnesyl. 2 farnesyls make squalene. We're up to 30 all of a sudden, 30 carbons. All right, and I'll go over this again next time, so relax. Squalene can rearrange 30 carbons into, look at this ring structure. That rearrangement is just twisting of bonds, makes something called Linosterol. This guy looks just like cholesterol. You'd think 1 or 2 steps to get to cholesterol. It's an additional 19 steps from here to cholesterol. I'll say more about that next time. I'm sorry? Yeah, they are, basically, yes.
Do you know about adipocytes?
Yep.
Their inner membrane is different. Do you know
It's a good question. You know, I don't know an awful lot about adipocytes. You would expect that they probably do have some differences there. They have to accommodate those. What we'll see in the body is that the body has some very cool tricks for solubilizing fat, because fat actually has to move from adipocytes out into the peripheral cells. It has to move. And so it's got some proteins that it does that for, and I assume that's what happens in adipocytes, but I honestly don't know if that's the case.